In order for a pathogen to be able to infect a host and actually survive long enough in order to cause disease, 7 steps must be taken and completed by the pathogen. Okay, so it's going to have to maintain a reservoir. It's going to have to be able to get into a new host, attach to the specific cells that it's going to get into, invade those cells or deeper tissues, and then it's going to use certain strategies called virulence factors to try and be successful colonizing the host. It's also going to deploy various evasion strategies to try to avoid our immune system, and then it's going to want to get out and infect a new host. So this video is mainly going to focus on step 1, which is maintaining a reservoir. So a reservoir is basically an entity. So it's going to be a person, an animal, or some sort of environment where the infectious agent or pathogen is going to be just normally live and replicate. Okay, so this is where I just kind of think of this as like their home, or they're kind of chilling out doing their thing. Okay, they don't cause a diseased state in their reservoir. Okay, they do that in a host. Okay, so at this point, this is where they're just naturally found when they're on their own. So there are 3 big categories of reservoirs. The first is going to be an animal reservoir. And so with these particular types of diseases, they are called zoonosis. And so a zoonosis is a human disease, right, that is caused by a pathogen that maintains an animal reservoir. And so in these instances, a lot of these diseases are transmitted from one animal to another animal, with humans actually just being an incidental, convenient host. Okay, so again, if you hear the term zoonosis, a lot of times that is actually going to be focusing on those diseases that are transmissible under normal conditions from a vertebrate animal to a human. Okay, and so the PowerPoint lists several different examples like rabies, which has a variety of animal hosts, bats, skunks, raccoons, etc. Lyme disease, the animal reservoir is actually a deer, and it actually gets transmitted from the deer via the tick to a human. So the tick is what we call a vector, but we'll talk more about that later. Okay, so animal reservoirs is just where the pathogen naturally can be found inside of an animal. Human reservoirs, so many infectious diseases actually have humans as their reservoir. And so in these instances, the disease can actually be passed from one individual to another without any kind of intermediary. And so for something like smallpox, right, where humans are the only reservoir, it turned out that smallpox was eradicated for a long time once we were able to have a vaccine against it, because the reservoir, it could no longer live and survive inside of the human reservoir. So when we think about human reservoirs, we often refer to this as carriers. And so there are 3 types of carriers that we're going to be talking about. An incubatory carrier is going to be an individual who has an infection and is able to transmit the pathogen during the incubation period. And so they haven't exhibited any signs or symptoms of disease. They probably have no clue that they're even infected, okay? But at this point, this is when the carrier is infecting other people, okay, during the incubation period. Another type of human carrier is going to be an asymptomatic carrier. And this is somebody who never experiences any signs or symptoms, never has any indication that they are actually infected. Okay, so they go whole shebang, where the organism is replicating, and they just never get sick, but they're still able to pass the pathogen on to another person who very well might exhibit signs and symptoms and hit that diseased state. The third type of carrier that we're going to talk about is the chronic carrier, and these individuals actually harbor the pathogen for months or even years after they are initially infected. So an example that we're going to talk about later in Unit 4 is actually going to be Typhoid Mary, who is a woman named Mary, who actually was a chronic carrier of the pathogen Salmonella typhi. And she was a chef in, I believe it was New York City. And so because she was a chronic carrier, had no idea that she harbored this pathogen for years. She accidentally infected dozens of people, and they got really sick, and it became quite problematic. But we'll talk more about that later. So for now, just know the difference between these three types of carriers. And then we also have non-living reservoirs, also called environmental reservoirs. Four of the most common are listed here. So soil, also plants, water, dust, food, all of these can serve as reservoirs for infectious agents. Later, we're going to talk about some different fungal pathogens, and you tend to find those in the soil. The bacteria that I studied in graduate school, which is Legionella pneumophila. It causes Legionnaire's disease. You can only get Legionnaire's disease by inhaling contaminated water droplets. So in that case, water is going to be the reservoir for Legionella pneumophila. Another one found in water is Vibrio cholera. And so that's when feces has actually contaminated a water source and people consume it. And with cholera, it's a pretty nasty disease. A lot of people get what's called rice water stool, and they actually lose several liters of fluid every day. So in third world countries, it's a leading cause of death in some of those countries, or at least a top cause of death. With food, one that we're going to talk about quite a bit later is botulism, which is caused by Clostridium botulinum. Remember, Clostridium is one of the genera that can form endospores. And so a lot of these endospores are naturally found in the GI tract of like livestock. And so as they go to the restroom, those endospores form in the soil. So you'll find Clostridium endospores sometimes in food, especially canned food, hence why commercial sterilization. There's now very strict policies on how to sterilize canned goods so that we don't get botulism when we eat those types of foods. And then another one in the soil is going to be the Clostridium tetani, which causes tetanus. Another one that's common to dust is the one on your PowerPoint, which is Mycobacterium tuberculosis. Remember, Mycobacterium has that mycolic acid, and so it's actually able to survive in like respiratory droplets and things, but it can also survive in dust because of that mycolic acid. So there's kind of start off as respiratory droplets, they can actually hang out in the air and things as dust, in dust, and be transmitted to a host. All right, y'all, that is going to be the end of this video. The next video is going to talk about step 2 in causing an infection or potentially a diseased state. Let me know if you have any questions.